Good morning, everybody. How are you doing? I hope well. Welcome to the final lesson for Unit 6, Biotechnology. This is Chapter 20. So let's define it first. Biotechnology defined as biological processes, organisms, cells, or cellular components that are exploited to develop new technologies. So we're going to take a look at some of the tools and techniques as well as the applications. And your job is to have a conceptual understanding of how these things work. So I'm going to be highlighting a few that are going to be most important for you. Okay, so first up, what's the relationship between science and technology? We're also going to look at the question of how have developments in genetics led to the developments of new technologies? What are the implications of genetic technologies for society? And the other question is what ethical and moral responsibilities do scientists actually have? That question we sort of looked at when I showed you the video on whether or not we should bring back the woolly mammoth, right? Just because we can do something doesn't mean we should. Okay, so first up, we're going to look at restriction enzymes and what they are, how they work. We'll look at vectors, what they are and how they work. And then we'll pop into some of the application of recombinant DNA technology that specifically has to do with restriction enzymes and vectors. Then we'll come back and look at the tools and techniques of electrophoresis, some sequencing and labeling, PCR, gene libraries, and microarrays, which we actually did an activity with a while back. If you remember, you got to act as oncologists, and we chose the correct treatments for the patients that had been diagnosed with breast cancer in class. Okay, so first up is restriction enzymes. What they are restriction endonucleases, right? Restriction enzymes. These enzymes only cut DNA at very specific sequences, hence the term restriction. You can think of them as chemical scissors. Okay, they cut usually in 4 to 8 base pair lengths at a restriction site on the DNA. And hundreds of them have been isolated. So they probably serve as a bacterial immune system against the phages. So I'll blow that up a little bit for you. And their discovery won a Nobel Prize in 1972. Okay, so here's a good example. This is EcoRI, and it cuts whenever it sees a GAA-TTC sequence in the genome. So whenever this shows up, that is where it will cut the DNA. So restriction enzymes, these weird names, they're named for the bacteria that they're isolated from. Different restriction enzymes recognize different restriction sites, so they're very specific. So this particular restriction enzyme is PVU-I, and you can see where it's cutting, right? So here's the sequence, the 5' 3' end, right? And it cuts in between these ends, and it leaves open what we call a sticky end, and we'll talk about that in a minute. All right, so DNA from different sources can be recombined by treatment with the same sticky end-making restriction enzyme. Ligase. So ligase is the molecular tape. It can be used to seal the break between the strands. And so we can create recombinant DNA from this, or DNA from 2 different species, which has great applications in biotechnology. So let's look at the diagram over here. We've got a restriction site. Here's our DNA molecule. We can see where it's cutting. So the restriction enzyme is going to cut the sugar phosphate backbones. And this leaves an opening here. So these are the sticky ends. A new DNA fragment can be inserted, right? Base pairing rules still apply. And so that will be inserted in. and then DNA ligase will seal that closed. And so down here, you now have a recombinant DNA molecule, or a DNA molecule that has two different species DNA in it. All right, so there are many types of restriction enzymes. So generally speaking, they recognize what's called a palindromic DNA sequence, right, or repeating DNA sequence. They can either cut in the middle of the sequence, so that would be a blunt cut, straight down through, or they can produce a 5' overhang of a few bases, and that's the sticky ends. And if we're going to do some biotech and we're going to create new DNA from two different species with recombinant DNA, it's the sticky ends that we actually want. All right, so let's take a quick look at vectors and how they work. And we're really going to concentrate on the plasmids, the circular DNA in bacteria, because it's very useful to us. DNA molecules can be modified to store and then replicate other DNA sequences. So examples of this are bacterial plasmids, bacteriophages, viruses, artificial chromosomes. And to be useful, the plasmids must have the origin of replication, a region containing lots of restriction sites, and genes that enable screening of cells, which we'll look a little bit more closely at when we talk about bacterial transformation. There is a size limit on plasmids. To get more genes into cells, artificial chromosomes can be used, and artificial chromosomes can act like a storage genomic library for us to store DNA in. Okay. Little close-up, right? So here's your bacterial cell. It's bacterial DNA. Here's our plasmids, also circular bacterial DNA, and DNA is double-stranded. That's why you see it that way. And so these plasmids are extremely useful for us because they can be cut with a restriction enzyme. We can insert new DNA to make recombinant DNA. And then we can insert that plasmid into any bacterial cell and it'll produce whatever protein that we want. Okay? All right, so I'm going to skip forward. We're going to pop down to some of the applications of how this works. And so we want to look at genetic engineering and some of this recombinant screening that we do with these restriction enzymes. All right, so first up, genetic engineering, combining genetic information from two or more organisms that can create a transgenic organism that expresses new traits that it didn't before. So bacteria are commonly used for this because we can manipulate them very easily. It involves the chemical scissors, the restriction enzymes, and then the vectors, right? And so the vector that we would use would be a plasmid vector. Okay, it's been accomplished in all domains of life, used in medicine, agriculture, industry, and research. And let's take a close look at how this works. Okay, so here's the bacterial cell. Here's its plasmid. Here is the DNA of a foreign chromosome. It contains a cell containing a gene of interest. So there are a number of genetic engineering techniques we have used to do this. Human insulin is a great example. We can make human growth hormone this way. We create the protein that dissolves blood clots in heart attack or stroke therapy. We can alter genes for bacteria that clean up toxic waste, and then we can insert genes for pest resistance into plants. And so this is the way it works. You take the plasmid out of the bacterial cell. You use restriction enzymes to cut the gene of interest out of a different cell, right? In this case, we can see it's a eukaryotic cell. We insert that gene in to the sticky ends. Okay, so now we have a plasmid that's recombinant. And then we place that plasmid into the bacterial cell. And then the host cell is grown in culture. So that means we're making lots of clones of the original bacterial cell. And this bacterial cell will now, with the gene of interest, create whatever protein. It will express whatever protein that we want it to. Okay, so it has lots of applications that I just went over below. We can harvest that protein. This has been tremendous for us. The discovery of the human gene for insulin. This is how insulin is made so that people with diabetes can actually have treatment with insulin injections as needed. Prior to that, there was no way to get it, and they actually used horse or pig insulin to treat diabetics, so you can imagine how well that worked out. So this has been a revolutionary biotech um, application that really helps for human health and for, for the wellness of people. All right, so I'm going to put a link up to this on my website. You can watch the making of recombinant molecules later on from DNALC. All right, let's look at that recombinant screening, what this means. So the frequency of successful engineering events can be low. And how do you know if that bacterial cell is actually expressing the engineered trait that you want it to? And so what you need is you have to have some reporter genes. These are genes in the vectors, like a plasmid, that enable detection of successful engineering. And a good example of this is ampicillin resistance. Ampicillin is an antibiotic, and so if the bacteria is resistant to the ampicillin, it will survive in an environment that has that antibiotic in it. Okay, so let's take a look again. Here's a bacterial plasmid removed. Right? We've inserted the ampicillin resistance gene into it, you see? So that's the reporter gene right there. And we've got a gene of interest from hummingbird DNA, okay? So we take out the plasmids, cut them with restriction enzymes. You can see the sticky ends right here. And then we insert the gene of interest in. So now you've got recombinant plasmids with the gene of interest from another species. Okay, you also can have a non-recombinant plasmid over here. We keep the original, right? Because we want to see if this actually works. And so when we insert these recombinant plasmids in, and we make copies of the non-recombinant plasmid, when you put them on a nutrient agar plate, the colony carrying non-recombinant plasmids will, with the intact gene, is not going to express whatever protein it is that you put in there, okay? The colony carrying recombinant plasmids with the disrupted gene can also be in there. And you see that there are two different colors. Okay, so often we use these fluorescent tags to actually show through the reporter genes whether or not our genetic engineering of recombinant DNA and bacteria worked. Okay, so a good example of this is doing a lab with the GFP expression of bacteria that identifies successful transformants, right? So this is a great example of bacterial transformation. And actually, when we had gone out of school on that Friday, this is the lab you would have been doing as a double period upon return. So we had wanted to do that with you. But I'm going to go through it. Bacterial transformation has a lab activity that I have designed for you. You'll do it in our next lesson. And let's go over what you should see. Okay, so there are 4 plates here. And we use Lauria broth, that's what the LB means. The nutrient agar that sits on the bottom of these Petri dishes has Lauria broth in it. So for our first one over here, okay, we can see on the outlines, if I outline this right here, we follow the cursor, this is a lawn of bacteria, right? So that means there's millions and millions and millions of bacteria that have grown. And we can see it as lawn, okay? So it's grown on the Lauria broth. There's no ampicillin in the nutrient agar to kill it, so it's thriving and growing. So this is considered one of our controls. Okay, then we've got over here, you can see the Petri dish with absolutely nothing in it. And why? Because it's got Lauria broth, but it's got ampicillin in it. So the antibiotic has killed all the bacteria. So we see nothing. It didn't grow. Okay, down on the bottom, when we add that vector, that plasmid, with the DNA of interest in it, we can see these white colonies. Okay, so this tells us in this one on the bottom left that when you've got the white colonies appearing here, they are resistant to the ampicillin, so they were able to survive. So that recombinant reporter gene is actually working. That's here. Okay, and it doesn't show up as lawn like that one because there is ampicillin in the nutrient agar in the Loria broth, right? You see that says it down on the bottom, LB-AMP. And so only the white colonies show up. Those are the bacteria that have the ampicillin-resistant gene. And then in our fourth plate here, we have a gene of interest. So in this case, it's the GFP, the gene that makes jellyfish glow. And so each of these colonies has the reporter gene for the ampicillin resistance. That's why they are shown up as colonies and not as one. And they've also taken up the plasmid that has the gene that make them glow. And so now the bacteria will glow. And so we can see our transformed bacteria here, and we know that it worked really well. And the ampicillin resistance, right, identifies all the bacteria that took in the plasmid, that vector carrying the gene of interest that we wanted. Okay, so down here on the bottom, these are both considered to be recombinant bacteria that are expressing a trait that we've manipulated. All right, so the development of GFP as a reporter gene has lots of applications outside of genetic engineering too. And here you can see the various fluorescent proteins showing gene expression for Drosophila or fruit fly development, right? And so these reporter genes are actually showing us what's developing where in the structure of the fruit fly, so it's very useful. Okay, we also can use complementary DNA. So these are DNA copies of RNA molecules, and they're used using they're produced, using the enzyme reverse transcriptase that we can take from our RNA viruses that have this enzyme. And so the complementary DNA increases stability. You can remove the introns for a prokaryotic expression of eukaryotic genes, right? So this is us. We want the prokaryote, the bacteria, to express some type of eukaryotic gene, and we can use complementary DNA for that. So with eukaryotic engineering, it is more difficult than dealing with making recombinant bacterial cells, right? And this is because of the nuclear membrane. And any time that you have to puncture a nuclear membrane, the odds are that you may be killing the cell. So there are a couple of techniques for it. So we can use a bacterial cell actually as a vector for this. You can see the recombinant DNA. In here, it's got down here, our gene of interest, and then we can insert that we can insert that into our plant genome, and you can get your results with that plant. So if you remember the video I showed you about RNA interference, this is a way that they did that particular technique. So we can use a gene gun to do it. That can shoot micropellets coated in DNA to try to get those pellets inside the nucleus. Right? And now we've got CRISPR. So this is relatively new discovery. The CRISPR-Cas9 protein complex is an enzyme. And basically the way that it works is you can target DNA. So the complex attaches to the matching DNA sequence, right? That would be the yellow sequence here. And then CRISPR-Cas9 complex will cut the double strands of DNA, and then you can add programmed DNA into that cut. So the idea of this is that, let's say somebody has a faulty gene early in development, then you would be able to excise that gene out and then using this enzyme, guide the appropriate DNA in to correct the genetic disease, whatever that may be, something like sickle cell. This doesn't exist for humans yet. It's very complex, but it's a good start and something new in biotech that's happening right now. All right, so if we talk about gene therapy, engineering cells in a multicellular organism can be very tricky because in order for something to stick, you have to remember all cells in your body have the same DNA except for your sex cells. So in order to really impact the DNA in the future for gene therapy, you mostly need to do it on an embryonic level because you can't change trillions of cells' DNA as once you're differentiated, once you're born. Okay, so it has to be done before those cells are actually differentiated. However, we have done some things. For example, here's a clone gene. You can insert it into a retrovirus, is the idea. There's a retrovirus capsid and use that as a vector to infect bone marrow cells that have actually been taken out of the patient, right? So this isn't done in a live patient's bone marrow, right? So we take this out, manipulate the DNA, and then the viral DNA will actually carry this allele inside the chromosome, and then you can take what's been done outside the lab and inject that into the patient in the form of new stem cells that will produce normal proteins in normal cells. That's the idea behind it. Okay, in complex engineering, this is multi-gene traits. It's become a little bit more common, so I'll show you a couple of examples. For example, this goat can produce human blood clotting protein in its milk. So we call this a coat farm animal, right? So we collect the goat's milk, and then they can extract the human blood clotting protein for medicinal treatments. And of course, this has been really great for humans. Golden rice is an engineered variety of rice that expresses beta carotene. That's a precursor to vitamin A. So in areas of the world where they may be lacking in vitamin A, which is essential for eyesight and some other things, golden rice can be engineered and it's much more nutritious, right? So here's a good example of a genetically modified food that's benefited 1 to 2 million people a year, and it helps to prevent vitamin A deficiency. And so they use a gene cassette to do this. Kind of neat, right? So that's how that's been done, and then it's inserted into the plant. Okay, so what we want to do is kind of pop back and look at some of the tools and techniques that are important for you to know. And the first one up is going to be gel electrophoresis. Many of you may have had the opportunity to do this in the past. It's a way to separate fragments of DNA based on their size, okay? So if you recall, the DNA is charged, and the phosphate group on the sugar phosphate backbone is negatively charged, okay? So when we expose the gel to an electric field, we can actually pull down towards the positive end the smaller fragments. So it will separate by size. The larger fragments will get stuck at the top. The smaller fragments move down to the bottom. And this gives us a very unique pattern between individuals. Okay, so DNA is placed into this matrix gel. Here's the gel. And it has wells in it, right? So those are just open spaces that we can use a micropipette and we can put our DNA in. And then once you attach this to a battery or you plug it in, then over time it will pull the smaller fragments down to the positive pole in the gel, and then that will leave a very specific pattern that we can see. And that pattern, right, each of these represents DNA sequences, shows us the DNA fingerprint or the unique banding pattern of DNA fragments. An actual gel has to be put under some light so that you can see those DNA bands. And so wherever it lights up, that tells us that DNA is in there, and then you can see the unique patterns that show up. All right, so labeling is the way to identify those sequences of interest. So I'll just show you how it works, right? And here's a great example. So this contains, in all gels, we actually use ladders on the side to tell us the size of the fragments, right? So this is a lambda. It's a restriction enzyme. Here's our this is our 1 kb ladder, right? It gives us a specific size, and then we can insert into the wells the DNA. And so here's DNA from Ted Bundy. This is found from the boot. This is the foyer. Here's O.J. Simpson, Nicole Brown, and Ron Goldman. And so what we can do is we can compare these DNA fragments to see what matches, okay? And so if we look at the boot here, and we compare it to the foyer, we can clearly see it's not a match, right? if we compare O.J. Simpson to the foyer, what are you seeing? You see any? You notice anything there? So we got a match. Here's Nicole Brown's DNA. That's going to be different. And here's Ron Goldman, also different. So this can be very useful for us in determining DNA fingerprints at crime scenes. It can also be very useful in determining paternity, or who's the daddy. All right, so DNA fragments are isolated. You have to separate the strands, and then those fragments are exposed to an oligonucleotide probe tagged with radioactivity for old or fluorescence for new. And all oligonucleotides are these 13 to 25 sequences. These are nucleotides designed to hybridize to DNA or RNA sequences and act as primers. And the bonds are a complementary sequence to the fragments. All right, so that's gel electrophoresis. We will do an activity with that in the next couple of lessons so that you can see how it works. All right, here's PCR, or polymerase chain reaction. It's a way to produce copies of DNA without the use of cells. This won a Nobel Prize in 1993. And an interesting story with it, Carrie Mullis, Dr. Mullis, very famous, used to surf a lot, live out of his van, and came up with the idea when he was out surfing one day. Won a Nobel Prize. All right, so how it works. A target DNA sequence is analyzed, right? So we only have a small segment. We need that primer to bracket the sequence, right? And so the bracketing of the sequence ensures that we're only going to amplify that particular sequence. The DNA primers, the primers, the free nucleotides, there are some coenzymes, and a special enzyme called Taq polymerase are put into a thermocycler. And then the thermocycler is programmed and it runs, and then the sequence can be copied 2 to the 30th times overnight, or actually, like even now, within a few hours, we can have a copy. So this is what a thermocycler looks like. This hatch will pop open, and you'll put your DNA samples inside. It's essentially a fancy oven, and it can move between temperatures very quickly. Taq polymerase, in order for a PCR to work, Taq polymerase is not destroyed at high temperatures. So it's isolated from a bacteria, an archaebacteria that's found around the hot springs of Yellowstone National Park. And this particular enzyme is not destroyed by high heat, so it makes it really useful for PCR. All right, so we're just going to go through the steps quickly so you can be familiar with it. In the first step, we have denaturation, right? So we can see our target sequence. You can see we're heating it up, and all that does is allow the strands to separate. Then the primers are added, and here's the primer, right? Remember, we're copying this DNA, and so in order for Taq polymerase to do this job, no, it's not DNA polymerase, it's Taq polymerase. In order for it to add the new nucleotides in, you have to have a primer there. Okay, so the annealing process is having the primers bond to the template strands, and that's done at a lower temperature. Then there's extension of those strands, 75 to 80 degrees Celsius. Taq polymerase is going to attach to the primers and replicate the target sequence. So this will happen over and over and over again. And each time that this is occurring, right, cycle 1 is going to yield 2 molecules, 2 will yield 4, 3, 8, then 16, and it looks like this.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, the process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
And that's PCR. All right, so let's just take a quick look at genetic testing using biotechnological tools to look for the presence or absence of particular genetic sequences. You need restriction enzymes for this. And then once you have that gene of interest, you need to amplify it with PCR. And then gel electrophoresis lets us know that we actually have the amplified sequences that we want. Okay, and so what we're typically analyzing are restriction fragment length polymorphisms, or RIFLEPs, right? And so you're looking at small fragments that when you put them through a gel, will organize from larger sequences to smaller. You can measure those and then you can compare and see what you've got. Okay, so this tells us about identity. We can look at areas of the genome that have many repeating sequences. These are called STRs, or short tandem repeats, and by comparing enough of these areas, you can confirm or rule out identity. So let's do a good example of this. This photo shows Mr. Washington just before his release in 2001 after 17 years of wrongful imprisonment. And so there was a rape committed. Here's the semen on the victim. And here's Earl Washington's DNA and STR data, and Kenneth Tinsley. And so if we look at the markers, here's the semen on the victim, 1719, 1316, 1212. If you look across at Earl Washington, you may have one STR that shows up, but not all, right? So we can actually see who the actual rapist and murderer was in this instance right here. Okay, Kenneth Tinsley was the one who murdered the victim. All right, in health, we look at genetic markers associated with various traits. Okay, so in this example, we can look at the RIFLEP, the restriction length polymorphism, due to a single nucleotide polymorphism leading to loss of a restriction site, and you can visualize that on a gel, right? And so here we have sickle cell again, right? You can see the normal protein here, and then in the sickle cell, right, we can see the mutant. So when you run that on a gel, you can see the differences between the two, right? So here's our RIFLEPs analyzing. Larger fragments get stuck at the top, smaller at the bottom, and we can tell the differences between the normal allele and the sickle allele, particularly down here, the smaller fragments. Okay, so the only limitation for genetic testing is experimental creativity, basically. So we use gel electrophoresis for lots of different things. Quickly on cloning, we're copying a genetic sequence. A single gene can be cloned. However, that's not really what we think about when we talk about cloning. So usually the term cloning, people are looking at reproductive cloning, a completely new organism. Cloning is actually normal. Plants clone themselves all the time. It's only controversial for organisms that don't reproduce asexually. Okay, so let's kind of pop through. Reproductive cloning produces an entirely new organism. We can do this with a plant. We have done it with animals, almost all kinds. And for animals, that's called somatic cell nuclear transfer, right? So here's the plants. Not quite as exciting as looking at how you do it with animals. So this is Dolly, the first cloned mammal. So the way that it works is you take a donor cell. In this case, it was a mammary cell. So here are the cultured mammary cells taken from this particular sheet. And then you take a different sheet, and if you note the different color in the faces, that's on purpose, the phenotypes. So you take an egg cell donor out of a different sheet, and you remove the nucleus entirely, right? Because you don't want the DNA from this sheet, and we want that sheep's DNA. And so then we take the nucleus out and we fuse these cells together. And so now inside here is the nucleus from this particular sheet from the enucleated egg cell from the egg cell donor. We grow this in culture, give it a little zap, make some early embryo. Then you can implant that into a third sheep. Why? Because we want to make sure it's not either of these two, into a surrogate mother. And then through embryonic development and birth, you eventually get Dolly, the lamb, who is identical to the first sheep over here. So this is, it seems easy. It is not. This took basically 365 tries to actually be able to clone Dolly successfully. So that means all of the other 364 embryos died first. Well, we have cloned cats. Here's Cece, the first cloned cat. We've cloned most things. However, mostly what we do with therapeutic cloning is taking specific cells and using it for medical purposes and treatment. And so to do this, you need some pluripotent stem cells from an early embryo. Those are stem cells that are not differentiated yet. Okay, so we can do an experiment. Here's a frog embryo. You've got a less differentiated cell. We can take the donor nucleus, transplant it in. Here's a frog tadpole here. And in either case, with the frog egg cell exposed to UVI, and you've got this enucleated egg cell, you can put the donor cell in. And so if the original cell from the embryo is less differentiated, right? I mean, it doesn't have a specific function, versus like an intestinal cell from a frog tadpole that you put in, it really requires the less differentiated stem cells for most of the clones to actually develop into tadpoles. Whereas if you have a differentiated cell, like from or more of an adult cell, it's going to stop developing, right? Because it already has a specific job to it. Okay, so we've done this with embryonic stem cells, and some of you may know people who've had stem cell treatment for injuries. We can take those embryonic stem cells that do not have a job, and the idea is that through culturing, you can actually create many different types of cells that can be used for gene therapy in the future. Okay, so the idea here would be here's a patient with damaged heart tissue or other disease. You remove skin cells because those are easy to get, and then you would reprogram the skin cells to become these stem cells, and then you treat them so that they differentiate, say, back into heart cells that can then be put into the patient. So that's the idea. It's not actually happening yet. We can only do it on with like stem cell treatment for plasma cells to reduce inflammation in joints. That's about as far as we've gotten. All right. So a few other things. Gene libraries are collections of DNA. There are sequences stored in vectors. We can take large DNA sequences, store them in plasmids or in bacterial artificial chromosomes, and it gives us a library where we can keep DNA, freeze it, and then use it as we see fit for biotech later on. Okay, so that would represent a genomic bacterial artificial chromosome gene library, right? And they're all recombinant plasmids. If we want to see what the sequencing is, it's a way for us to determine the sequence of bases for DNA, and that won a Nobel Prize in 1980. So I'm just going to show you how it works pretty quickly. The Sanger method was how I learned it in school. Now it's a whole lot easier for us to sequence DNA. It's replicated in the presence of what we call dideoxynucleotides. So these are nucleotides that have a small piece removed in them. When it's incorporated, it's actually going to terminate replication because there's nothing else that can bond to it. Okay, so I'll show you how it works in just a moment with a quick video, right? But basically, anytime this dideoxynucleotide is added here, it will literally stop the replication at a certain point. And then we can work backwards to see what the actual sequence of the DNA is, okay? So the old method, this is the old school method, where you'd have these printouts that were really, really, really long, and you had to work your way backwards. Thankfully, this is not something you have to do anymore. Um, the cost for actually doing this has gone down tremendously. A couple $100 and you can have your DNA sequence. So let's take a quick look at Sanger sequencing.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into two strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all four bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
Okay, so that's the Sanger sequencing method. All right, and then microarrays, we actually did this before. That's the complementary sequence. We can isolate the mRNA, create some complementary DNA, wash it over, and then we've used this in applications such as like we did in lab, trying to figure out, if a particular cancer gene is being expressed or not, and then what type of treatments you have from it. So since we did that in class already, I'm going to skip over this for now. All right, so those are some of the tools and techniques. You're going to be responsible for knowing how restriction enzymes work, what a vector is, how gel electrophoresis works, what DNA sequencing is, and PCR. Okay, and then we went through some of the applications here of what genetic engineering is used for, right? And so just make sure you can identify some tools and techniques that we just discussed, explain all the applications, and then ethics we're going to put to the side for now. It's very interesting, but in the interest of time, we're not going to do that. All right, so I hope that's helpful, everybody. Let me know if you have any questions, hit me up, and we'll do some activities to help you understand biotech a little bit better. See you later.